Good evening, students.
Good evening, sir.
Good evening, sir.
Good evening. Okay. Right. So, I think in yesterday's class we were talking about the components of genetic markers, right? So we discussed a few genetic markers yesterday. I think we were talking about RH and HLA, and as well as haptoglobin. We were talking about, correct? Okay. Let's continue from there. So in the last part yesterday, we were talking about the haptoglobin. So briefly remember what is haptoglobin, what type of protein or a component it is. And then you can focus on understanding the polymorphism related to that and any applications of polymorphism with respect to that particular protein. Okay. So we talked about haptoglobin and haptoglobin being the hemoglobin carrier. Okay, basically allowing the loss of hemoglobin through the kidneys, which can damage the kidney portion. Correct. So this component we talked about, and polymorphism wise, we discussed that it has single gene, so two alleles. But even with two alleles, like any other normal allele combination, three possible outcomes. Correct. So which can be differentiated using a method like electrophoresis method. So what is the use in knowing whether someone has HP11 or HP22 or H heterozygous condition for 1 and 2? I was telling you many studies were talking about one racial differentiation or population-wise differentiation that have been noticed here. Okay, so people with in Southeast Asia and to Europe and Africa and to US, we said HP1 allele number increases, their incidence rate increases. Okay, so we also briefly talked about the percentages of HP1 allele being high in South America and being low in Asia, including India. Correct. So variation within the haptoglobin levels can be used as a marker, is a means to differentiate populations. Okay, so this should be the one of the key function or application of genetic markers, population level studies. Okay, and then age-wise differences are also noticed that serum concentration of this changes with age, starting with the 3 months. This is when it's starting to detect, and then it reaches the maximum at 20 years of age. So like that, age-wise difference can also be identified with respect to haptoglobin. And also haptoglobin, given that it has connection to the hemoglobin levels, one can detect the anemia caused mainly because of destruction of RBC. So the nature of anemia, because anemia can be because of multiple different reasons. But if it is because of destruction of RBC, which is known as hemolytic anemia, so destroying the RBC leads to anemic condition. That can be detected using haptoglobin levels. Okay, right. Also, there is a connection to heart attacks, rheumatoid diseases, diabetic nephropathy, or diabetic induced kidney damage. So those components can also be studied using haptoglobin. So these are some applications. And also I briefly said studies because some of these studies are not very conclusive. They might have picked up a few populations and counted the relationship between haptoglobin levels and their disease condition. Such disease conditions are showing some connection to HIV, high mortality rate with certain form of the haptoglobin. HP11 seems to show higher mortality. Similarly, malaria, particular form of malaria called falciparum or brain malaria, malaria that affects the brain function, which is the most dangerous form of the malaria, which is more common in Africa, less common in India. Such falciparum malaria also seems to be associated with HP11, so homozygous condition for that, and also some connection to the malignancies, various different types of cancers to the haptoglobin levels. So these connections, if we actually properly figure out, will help us to use the haptoglobin polymorphism in connection with the disease condition. So it can help us studying the populations, especially the diagnostics can be done here. So prevention of the certain deadly diseases is possible with the help of these type of haptoglobins. Correct? So keep in mind, in general, genetic markers, what are we learning about? Population studies, variation with respect to the disease incidence, severity of the disease. So, or we talked about even paternity dispute, solving paternity disputes. So these should be the major benefits or applications of genetic markers, and we're talking about various examples under them. Okay, so remembering factual data will be challenging, but develop the general idea, have some basic information figured out related to each of these components. As they are not frequently given in the exam, you're all right because you don't have to too much stress on by hearting these things. But have the general idea, make a summary of each of these components. That should be sufficient. Don't worry too much over that. But genetic markers, we look into the questions. I think I briefly mentioned the questions that have come in the exam, okay, in the past. But we are also going to briefly revisit them. But mostly ABO blood groups, RH, HLA. So these are the ones that are frequently asked in the exam. Few other topics I think are going to be covered in tomorrow's class and maybe partly towards the end of today's class have also been asked in the exam. So focus more on them. Okay, right. So today we are going to first begin with the transferrin molecule. So what is transferrin? So transferrin is another plasma protein. So plasma is the part of the blood, right? So serum, plasma that we talk about, we are referring to the part where proteins are present. In addition to the cells in the blood, we also have other dissolved molecules like proteins. So transferrin, look at the name. Previously it was haptoglobin. Okay, it's a type of globin or globular protein like hemoglobin. Here it is transferrin. So ferrin means what? Ferrin refers to iron. Trans refers to transporting iron. So transferrin is an iron transporting protein found in the blood or in the plasma portion of the blood. So what is transferrin? Iron transporting protein. And where is it synthesized? It is synthesized also in the liver. It is normally synthesized in the liver, and its job is to transport iron. Okay, transports iron. So from where do we get iron? Iron is normally received in the form of diet. So iron rich foods nowadays, I think Indian government is also emphasizing so much on taking iron rich foods, especially those people who are vulnerable to iron loss and the iron associated molecules. Where is iron so important for us? What is the key use of iron in our body?
Capturing oxygen.
That is?
In hemoglobin.
Basically, iron is integral component of the hemoglobin. Hemoglobin is integral component of the RBC. For RBCs to be synthesized, you have to provide enough iron. Correct? And women especially are vulnerable to the iron deficiency. Okay, because of their physiological system, they tend to lose more RBCs and they are in need of more iron-rich foods. That is why Indian government, as part of National Nutrition Mission, is promoting taking more iron-rich foods. Okay, anemia is a common problem in children as well as in women. Correct? And within the body, when we eat, when we take iron in the form of diet, what happens? Iron has to be carried by or taken by a particular protein. There is a carrier for that iron, which is known as transferrin. and especially in a particular ionic form called ferric form, ferric iron, or Fe3 plus, because iron can exist as Fe2 plus or Fe3 plus. So ferric iron, ferric version instead of ferrous version of the iron. So here, ferric version of the iron is bound to this particular transferrin. Okay. So what does it do? From the intestines, from the digestive system where iron is available, because from the food we eat, iron-rich components, iron gets released in the digestive system, and when iron is released into the intestines, that is where transferrin is available. Okay, so this is a common source where iron is found, to which transferrin normally binds to. And also where RBC gets lysed, like in the liver, like in the spleen, these parts of our body, what do? We recycle RBCs. In the process, we also release hemoglobin and iron. We also have transferrin being present in those regions to capture the iron. From there, wherever the iron is rich, like the intestines, digestive system, and the hemoglobin breakdown like liver and spleen, iron is captured by the transferrin and carries to wherever iron is mostly needed. Where do we need iron? Wherever RBC is synthesized. Correct? Where is RBC synthesized? RBC is synthesized in the bone
Bone
marrow,
marrow.
the soft part of the bone, right? This is where it takes the iron. So transport them to iron requiring cells like bone marrow and to the sites like liver where iron gets stored. So this is a job of transferrin. So this is a basic physiological role of the transferrin. Okay. A basic idea is enough here. You don't have to draw these complex structures. If you are interested, I am also going to show you another flow chart. What is transferrin? What is its role? This should be the subheadings to begin with. Transferrin is iron transporting protein found in the plasma, made in liver. Use of it. As the name suggests, it transports iron from where to where? From sites where iron is available, like digestive system, intestines, and wherever RBC gets recycled, to wherever iron is needed, to the bone marrow or to the liver, like that. Okay, and a free protein, free transferrin is known as apoprotein. APO in biochemistry, we normally talk about this, where apo means free form of the protein, not bound to transferrin. And holotransferrin is a version of this where iron is bound to it. So apo is a free form of transferrin. Okay, and holotransferrin is where iron is bound to it. Okay. So like that, when iron bound to the transferrin, transferrin specific receptors will be there on the cells. On top of the cells, there are specific entry points for transferrin. So transferrin will react with these transferrin receptors and thereby taking the iron into the cells. Like that, transferrin carries the iron. In this flowchart also, don't have to worry about the specifics. The overall idea is important here. So we eat enough iron. We ingest iron almost 20 milligrams per day. They say it enters into the body. Some of it enters into the body. Some of it gets excreted in the body. Okay, and then this apoferritin, or free form of the iron, will be bound to ferritin while bind to the iron storage protein or iron transport protein. Okay, and then what happens? Then basically this the iron rich transferrin proteins. So iron rich transferrin protein will carry it, as we are talking about. So this is synthesized in the liver. So it will make iron soluble and it will prevent any iron, because if you leave iron as it is, iron can result in some amount of toxicity in the body. So it will also prevent that, because now iron is captured by the transferrin. So it will not allow the iron to cause damage to our body tissues. Okay, and also it carries the iron around to the cells where it is needed. So this is the basic idea. You can write like the flow chart here, or you can show it like this. Iron from the digested parts to the transferrin will bind and it will be carried to the cells. Okay, this is the basic idea and the function of the transferrin. Now let's focus on the polymorphism part of it. So what do we know about polymorphism of it? So does it exist in different forms? How many forms does it exist? And what type of polymorphic nature does transferrin show? Okay. So genetic variation in humans with respect to transferrin was shown back in 1957 by Smithies. A scientist called Smithies, he showed the variation among humans in transferrin protein. So another source for variation that they could find, only one gene is present. There's only single gene for transferrin. Unlike the cases that we have talked about HLA. So there's only one gene, single gene, but different variants. And interestingly, the variants are variants that they detected to show different behavior when they separate them using certain techniques. So they call them B.C.D. So the single TF gene shows three variations. What are they? B, C, and D. It's not allelic forms, but the form which behaves differently in the laboratory conditions, meaning some nature of it is different. Correct? So make them to get attracted to anode. Some of them get attracted to anode. Some of them get attracted to cathode. So it could be because of the difference in the amino acid sequences. That is where some of them get attracted to the anode, some of them get attracted to the cathodes. So positively charged electrodes, negatively charged electrodes, they get attracted. B and D, another one is called the common one. So like that, B, C, D, three variants. So you write like this: polymorphism here, B form, C form, and D form. B and D are anode and cathode form. C is a common form. Okay, the way they are showing the polymorphism is different here, as you notice. It's not necessarily the genes and allelic forms. Okay, and these forms, they've also noticed in literature, if you look into, some of them are more common in some populations and others are more common in other populations. So population wide variation can be noticed here. What is that? So write down these points. So Caucasians are people who are European origin, and in general sense, if you go to Americas, they are using the word Caucasians. They are referring to whites in America in a very informal sense, or people who are of European origin or Caucasians. In them, allele C, so or TFC, is commonly found. So what is more common in the Caucasians is the TFC form, transferrin C form in Caucasians. And then West Africa and the western stream of Bantu are, these are the group of ethnic group in African region who speak the language called Bantu language. So these people, so in Western Africa and western stream of Bantu, they have higher frequency of the D version of the TF. So C is more common in Caucasians and Africans, especially those who speak Bantu language. In them, TFD version is more common. And what about blacks in the US then? Logically. So what do you think it will be more common in the blacks found in the US? If Caucasians have more of C and Africans have more of D, US blacks will have combinations of both. Intermixing has happened in the US, right? So CD form has been most commonly. Okay, not necessarily most commonly. I think they show that it should depend on the intermixing pattern as well. About 10% of the US blacks show CD, heterozygous condition for the CD. And Australian Aboriginals, there are some studies that have indicated that they contain some amount of D, but not exclusively. So it looks like D seems to be, I think we have talked about in one scenario where like in Bantu areas where D is more common, but D seems to be not very well selected otherwise. So there may be some form of disadvantage of carrying TFD allele. Okay, but otherwise C is more common in Caucasians, in Western Africa and other regions is D. US blacks about 10% of the CD. Okay, so this is a bit about, this is giving us an idea about some sort of variation that you can see based on the transference with respect to different populations. Correct? Anything else is no. This is a polymorphism part. What is a polymorphism nature for transference? and any population-wide variation that we are able to see here. And next we'll talk about this is something really specific. Okay, you won't have to write too much about it because I was trying to find any connection with respect to population-wide studies on this. So it's up to you, but I will give you a little idea. If you can follow this, make a note of few points on this. Okay, make a note of few points related to this. So scientists have also found TF polymorphism and its relationship with the iron metabolism, how iron is maintained in the body. Okay, so if you see clinical correlations also can be seen here, and few aspects of iron related issues, like people with iron deficiency, anemia, where people don't receive or get enough iron, and anemia is because of that, because not enough RBC is formed, especially the cases with pregnancies. How do you notice them? So transferrin levels are high. So free transferrin level is high. Apotransferrin levels will be high. So apotransferrin levels can be of use, right? We want to identify, like the previous case, we found out whether hemolytic anemia has happened here, iron deficiency anemia. Not enough RBCs are formed because the raw material required for RBC formation is not available. How do you know? One way to check it is testing the transferrin levels. Free transferrin levels in the serum, in the blood plasma, can provide us a link on this.
So more transferring means iron deficiency.
More free form of transferrin means iron deficiency. Because if there is enough, there is a range for it. I excluded that information, but I remember seeing that in the clinical testings, they have percentages. If you have a think about anywhere between 20 to 40, if I'm not wrong, where it is kind of saturated. Below 20% of it, meaning we have more free iron, transferrin, less iron, and then enough iron to bind to the transferrin. Then if you have, too much of over percentage of iron, too much of saturation of iron is also bothered. Meaning how iron is maintained or present in the blood, you cannot directly say it, right? So indirectly, they would check the levels of transferrin. If there is more free form of iron, transferrin, it means there is less iron. Not much iron that can bind to transferrin. So transferrin levels can be used to study the iron deficiency anemia or the cause for anemia. Also, not only iron deficiency, transferrin can also provide us idea about iron overload. I think in some populations, there is a concept of iron overload happens. There is too much of iron intake happens in few populations. Why is that? I think one component I remember reading is in Africa, where people try to cook foods in the iron utensils. Okay, in certain tribal regions and some places in Africa, they cook the foods in the iron containers. What happens? These are not anodized, so these are not coated with anything. So what happens? Iron enters into the food. So these people are overloaded with iron. So sometimes even iron overload happens. Scientists have studied the relationship between this transferrin and the iron overload. And a particular polymorphic form of the TF, transferrin, also is studied here. Okay, so if you want, you can just leave it there. Say iron overload has been noticed in some populations, like in Africa, where cooking is done in the iron containers, uncoated iron containers. Uncoated iron containers. So for them, looks like TFCD heterozygous combination seems to be protecting them. So some people actually kind of showing resistance to iron overload when they have TFCD, heterozygous version of the TF, transferrin C and D alleles. So they seem to have some protection. Okay, I'm going to skip the other one here because it doesn't actually conclusively talk anything. So I'll just briefly mention, this is enough for us to write. If you have to discuss something like this, what is the polymorphic nature of it? How is that useful? Because this is sufficient. Okay, there's not much conclusion that I've seen here, so ignore these points. We'll not talk about them. We'll also just talk about this. TFCD has some protection to it. Okay. And I wanted to add, I was actually quickly checking if there's anything new that came on this part of its relevance to the diseases. Okay, iron and metabolism we talked about, but any known disease. And interestingly, I found a study that was done in India in 2019. This is published in Journal of Entomology and Zoology Studies.
Sir,
Yes.
yeah, like we have seen that more iron has been TFCD protects them from iron,
One,
overload of iron. So is it balanced for polymorphism again?
I don't know whether we can say it. I think it hints at something like balanced polymorphism, because where heterozygous condition is maintained, it could be, but I don't think we have enough knowledge about it, enough in-depth analysis. So don't use it as an example, don't cite it as an example, but it may look like that because you're maintaining the heterozygous condition there. So it can be balanced polymorphism, but more further studies are needed on it. Okay, we have very well-known, well-established studies for balanced polymorphism. Use them, but this could be also an example for that, which I don't prefer to cite it because it may not be well known, may not be commonly known example, right? So yes, what did I found? I wanted to add few points to our discussion from this. So transferrin polymorphism and its clinical applications. The study had come very recently. They talk about these TF versions and more details that we don't need. But basically those BCD versions I talked about, like BCD. Transferrin has three major isotypes forms, BCD forms. And most of us actually have C allele. And then some susceptibility to the diseases and polymorphism is seen. So some link between a disease condition and the transferrin polymorphism is seen. Let's make few points on this. So one point you can write here, transferrin seems to be important for the growth and differentiation activities. For our normal growth, we need transferrin, of course, because transferrin is connected to iron, right? And RBCs play an important role. So directly or indirectly, so transferrin plays an important role. I'm not sure if you're able to read it. This part I'm talking about. An important role for growth and differentiation activities. Differentiation means one cell getting converted into another tissue or becoming functional from the preliminary form. So in a way, we can call it as a growth factor because it influences the growth of our tissues. So it can be called as a growth factor. Okay, second point I want you to write is its antimicrobial activity. So this transferrin also shows antimicrobial, meaning it can kill pathogens, it can kill microorganisms. Generally, they thought it could be because iron is important for bacterial growth. So if transferrin is there, what will it do? It will capture the iron. Then bacteria will not receive enough iron. So thereby limiting the nutrients required for the bacteria to survive. So
Can you repeat the points? What we could write?
yes, I will. So I'm showing this here as well. Antimicrobial activity. The antimicrobial activity of apotransferrin. So it has antimicrobial activity, meaning it can kill microorganisms. How? The common understanding is it reduces the free iron levels, which are requirement for bacteria to grow or microorganisms to grow. So by limiting the iron availability for bacterial growth or microorganisms. So it reduces the iron levels available for the microorganisms to grow. Also, other mechanisms seems to be there, but otherwise what is important here is antimicrobial activity.
So does virus also need iron?
I don't think so, because virus is just the component where from genes it makes proteins. So it may not need iron because it doesn't have metabolic activities, right? It utilizes our own components, so make the proteins, spare parts, and goes up. So it should be the microorganisms like bacteria, protozoans, etc., not necessarily the virus. Okay, and also cardiovascular diseases. With respect to some cardiovascular diseases, okay, so the transferrin level seems to also affect the cardiovascular heart related issues or problems. Okay, it has some correlation, its levels have some correlation to the heart related disorders or cardiovascular problems or diseases. Okay, and the next point, the last point I would like to tell you is cancer therapy. So apotransferrin, or the free form of the transferrin, seems to also have a role in cancer treatments. Okay, cancer treatments. Okay, right. So I don't think we need more details than this. So some clinical use is there of transference. Okay, and also polymorphic nature. So we looked into what is transferrin, what is its role, polymorphism in it, and a brief applications of this component. Okay, I think that should be more than enough. We don't need more details about this. Okay, so that is about transferrin. In the syllabus, it is spelled wrong. It says transferring. It's not transferring. It is transferrin. Okay, next we'll talk about blood enzymes. We have GM and blood enzymes here. So first I'll talk about blood enzymes. Okay, I've taken an example here. So enzymes that are routinely found in the blood in a way. So the previous case of plasma can also be used as an example for that, but a specific example I wanted to take here. So which is G6PD or glucose 6-phosphate dehydrogenase. Blood enzyme called glucose 6-phosphate dehydrogenase. So glucose 6-phosphate dehydrogenase is an example for blood enzyme. Well, this is one of the blood enzyme, and usually in the physical anthropology, genetics part of the physical anthropology, we hear G6PD here and there. I'll tell you where in connection, but first let me tell you briefly what is this glucose 6-phosphate dehydrogenase. As the name suggests, it has the suffix to this is ase, protease, nuclease, like that it's an enzyme. So G6PD, in short, or glucose 6-phosphate dehydrogenase, is an enzyme coded by a gene present on X chromosome. So G6PD is an enzyme coded by corresponding gene present on the or located on the X chromosome. So what is its function? So G6PD coded by a gene present on the X chromosome Okay, it is present in almost all cells of our body and important in processing carbohydrates. Sugars that we normally take in processing them, in metabolizing them, we need G6PD enzyme. The product of this G6PD gene present in almost all cells, okay, and responsible are involved in processing of sugars or carbohydrates. Okay, especially it is very important in RBCs. Excuse me. RBCs, we know that play a very important role carrying the oxygen from the lungs to the various parts of the body, right, as part of the blood. So RBCs are actually protected by this enzyme. So some amount of protection is given by this enzyme, especially some amount of damage can happen or destruction can happen, and which can be addressed by glucose 6-phosphate dehydrogenase. So it helps in protection of RBCs, which play an important role in the body. Okay, there's something more specific. I think I want to ignore this component here. A specific step where glucose 6-phosphate dehydrogenase is needed. So we have something called pentose phosphate pathway. Okay, but don't worry, this will be too much for you. So when glucose gets converted into other forms, this is where we need this G6PD. But in connection with processing of carbohydrates is sufficient for this. Okay, now polymorphism part. Let's look into the polymorphism because I briefly told you what is G6PD, what is its function. Now, in what ways we're talking about polymorphisms here? G6PD polymorphism is a different way that we explain it. Is either some people have a normal or functional G6PD or they're deficient for G6PD. So two categories of people, those who have G6PD active, those who don't have it. So people who have functional G6PD in them and who don't have it. So two forms. So it is not allelic forms. This is not the forms that are found in experiment. So this is having it or not having it. So people who are normal to G6PD and people who are deficient in G6PD. So in what form the disease actually goes or inherits G6PD? How does it inherit?
Either mother or father.
X-linked. So it's present on X chromosome, right? Either from mother or father. X-linked disorder. It's an X-linked disorder. Inherited genetic disease. This is inherited disease. Okay, where hemolytic anemia is seen. So people with G6PD, so RBCs cannot be protected. Normal carbohydrate metabolism, which is also important for RBC, is affected. Because of that, what happens? Anemia, blood loss, RBC gets decreased. So hemolytic anemia happens in them because they're deficient in this. Normally, abnormal RBCs like this are degraded. So it's not about the production of them, but it's about the destruction of them that will be high. So inherited disease called G6PD deficiency happens, which leads to hemolytic anemia. And this is a very common problem in humans, very common problem. So as per the old reports, at least 200 million individuals in the world have this problem. 200 million individuals have this problem. Especially in the Middle East and Africa, tropical part of Africa and Asia. Okay, this is more common. And this problem is because of several mutations, not one. There can be different mutations causing in G6PD deficiency, caused by about 400 different mutations, not all together, 400 different possibilities in a way. So G6PD gene can carry several different mutations and they can result in the deficiency of this product. So this is like severity changes based on what mutation it is, okay? And the connection with which, so 400 different mutations can lead to different forms of or different extent of G6PD deficiency. Where did we learn about G6PD? I'm not sure if I specified this, but people with malaria, where there is more malaria, okay, very much like how sickle cell anemia protects it, like balanced polymorphism we talked about. So G6PD can be also an example for that. So there, sickle cell anemia, abnormal shape of the RBCs provides protection. Here, G6PD deficiency of RBC provides protection against malaria. So people who are G6PD deficient, they also show some protection against malaria. So tropical places, tropical Africa and Asia could be, this balanced polymorphism is maintained because of that reason. So if it's an X-linked disorder like this, who are most affected, males or females?
Females.
Both.
Both. X-linked, who is mostly affected, males or females? Options are females, males, both, none of the above. Veda went with females and Sunil went with both.
Both.
So we have two more options left. Vishal, do you want to go with another one or do you agree with one of them? Preeti, Vishal.
Sir, it can also be someone with XXY.
It's a rare condition.
That they have more chances.
We're asking who is mostly affected, which gender is mostly affected because of X-linked disorder, and here we are talking about X-linked recessive disorder.
Males.
Hemophilia, who is mostly affected, males or females? Females can be carriers or affected. They have two possibilities. Males have no such possibilities. If they receive one defective X from the mother, that's it. They will be deficient for G6PD. So males are mostly affected because males only have one X chromosome. They don't have another compensatory X chromosome to make up for the missing protein. This is a logic I want. Okay, right, Shweta, did you get it? Okay. So here males are mostly affected because males only carry one X chromosome. If that happens to be defective, and that's it. They are mutant for that. Are you able to follow this, Preeti?
Yes, sir.
Good. What's your background, by the way?
It's a biochemistry honors.
What is it, sorry?
Biochemistry.
Biochemistry. Very good. So it must be easy for you, right? So G6PD. Have you
Yes,
studied
I
this?
already studied all this.
Very good. Yes, because this is a
Just
very
like
good
a revision for me.
biochemistry, right? Very good example from biochemistry because enzyme studies.
Yes, sir.
Good. So feel free to contribute in the class. If you find something that you can add, do that because we have people from different backgrounds. If you recognize there's something that can be added to what we are discussing in the class, feel free to do that. Okay. And no issue. Even if you are wrong, that's all right. I know it, I'll correct it. Okay? Otherwise, we'll add that to the information that we're discussing in the class. Okay.
Okay.
So G6PD deficiency like sickle cell anemia can be an example for balanced polymorphism. Okay, I don't think we need this. Yes. So deficiency, there's also variation depending on how active is the enzyme, because several mutations are there, right? So people may have some severe forms of the G6PD deficiencies, or moderate form of it, or mild form of it. Okay, so there is a lot of variation that can be seen in given populations. And the first two categories are, in a way, you can club it together with slight variation. So where below 10% of activity. So how much enzyme is active? Depending on the mutation rate, sometimes if below 10% of the enzyme is active, then what happens? Not much activity is there. So the deficiency is really severe. And such 10% and below severity form of G6PD deficiency is seen in Asians and Mediterraneans. Okay, so this especially this form of it. So where 10%, below 10% of enzyme activity is seen, so meaning it should be more severe in terms of deficiency. Where is it seen? Asian and Mediterranean populations normally show this. Okay, second category I wanted to make a note of is moderate form of deficiency. Moderate form of deficiency where about 10% and up to 60%. 10 to 60% enzyme activity is seen in them. So example, 10% of the black males in the US have this form of the deficiency. 10 to 60% activity is there. So about 50% or more activity can be seen here. Okay, and 10% of the black males have this version. Okay, third variety that we'll talk about here is mild form of it or no problem at all. So 60 to 150% enzyme activity, which is weird, seeing more than 100% activity, but all depends on what is taken as a reference point. In biochemistry, normally I think it's measuring the enzyme activities requires some form of reference. So I feel here the reference, it goes beyond the reference points, 1.5 times better than the reference. So better. So more than 60% activity to 100%, or because the reference more than that. So not much severity is there. So these people are usually normal. Okay, and they also have a condition that goes even beyond this. But this is enough. Severe form, moderate form, mild to none form is enough. You mentioned about at least 3 possible categories here, where deficiency varies, the effect also varies, and population-wide variation can also be seen to some extent here. Okay, right. So yes, I think we'll not talk about this drug. Some drugs I can together in combination with the G6PD deficiency can make it more difficult. But okay, I think we will skip that component. Okay, so let's learn a little bit more about the polymorphism part of it. So as I was telling you, there are many mutations that can lead to it. So I think we already talked about it, about a few hundreds of them. So different sources talks different numbers for this. But overall, there are several mutations that can lead to these problems. Okay. So, and also we have noticed that some form of, a relationship in distribution of these alleles. Especially, I was telling you, you can see a correlation between incidence of malaria and G6PD deficiency. Very much like what we talked about sickle cell anemia. So there is higher incidence of G6PD deficiency in correlation with wherever the higher Malayan incidence is seen. I think I was already indicating that to you before. So geographical distribution correlates with the malaria, presence of malaria. So, and we can use this because G6PD deficiency, as I was saying, can be used in epidemiological studies. So disease spread like malarial disease. So they can be used in understanding the epidemiological components, meaning disease origin and disease spread rate. We have dedicated a unit for epidemiological anthropology. So there you can give some examples like this as epidemiological studies. But here we are going in the genetic markers view. So particular genetic marker called G6PD shows variation among population, especially the regions where this becomes more interesting when we are studying them where malaria is common. Like for example, in Mali, in Africa, where malaria is a major problem, so disease susceptibility seen being different in two particular ethnic groups. The two groups called Fulani and Dogon. I was looking into a study on this, where we are seeing the example of use of G6PD here in Mali, where they have noticed that two ethnic groups called Fulani and Dogon, two ethnic groups called Fulani and Dogon, they both live and share same socio-cultural factors. They have same socio-cultural factors, and also they have same amount of malarial incidence. Malaria is equally common there, and they also have same socio-cultural factors, but they're affected differently. They're affected differently by malaria. So meaning their fatality rate, death rate seems to be different. So especially Fulani individual seems to be less affected by malaria. That could be because variation in the G6PD deficiency versions. So they have same amount of malarial incidence, they share same socio-cultural factors. So it could be more genetic factors that are causing the problem, causing the variation here, which could be G6PD deficiency. So population level differences with respect to or in response to disease can be studied using this type of markers. Don't worry, I'm going to skip this part. I was reading about this where certain mutations are more common, or certain mutations are found to be a certain percentage. Okay, and some correlation do you see? So a particular mutation called 202A, meaning particular position on the protein. So if you keep counting the amino acids, position 202, some mutation is there. This mutation tends to be 7.5% in severe form and 11% in the less severe form. Like that, some correlation can be seen between some varieties of G6PD deficiencies and the disease burden rate or disease severe. Don't worry about the number, but just mention this. The difference between these two groups, ethnic groups, could be because of the specific form of mutations that may be present. Okay, all right. So yes, I think we don't need to know about G6PD deficiency, how this can be managed. There are different ways, but with respect to the G6PD deficiency, so this disease correlation can be seen here with malaria. Okay, right. So that's briefly about the what is G6PD. This we took as an example for blood enzymes. You can also take the other enzymes as I was telling you as blood enzymes. Otherwise, I added one more example here, that is G6PD. So in relationship with the malaria, you may study this as an example. Okay, right. So now I would like to move on to GM. GM. So what does GM stands for? So GM markers. So GM markers refers to a specific form of antibodies. So in immunogenetics part, we briefly discussed this. So GM refers to gamma globulin markers. Remember, we talked about different types of antibodies, IgA, and we talked about IgD, IgE, IgG, IgM, IgG. Out of those, IgG is gamma globulin it is called. So we're talking about the variety or polymorphism with respect to a particular class of immunoglobulin or particular class of antibodies called IgG. So human antibody is immunoglobulin G. It's gamma globulins. Okay, but in generally, even before this definition of IgG, so initially scientists thought they called them immunoglobulins, and they in general they call them gamma globulins because in the electrophoresis process, like separation of those DNA sequences, it moves to a specific location called gamma region on what is called a paper electrophoresis. So experimentally, when they found out, so for historical reasons, it went to a region called gamma region, and it is called gamma globule. So we are referring to what here? Immunoglobulins. And within particularly this IgG, so scientists have noticed, someone called Grubb has noticed allotypy within IgG. So what does it mean? So he noticed some forms, varieties or polymorphism with respect to IgG, which is known as allotypy. So what does allotypy means? Allotypy refers to small differences in amino acid sequences in few of those antibody structures. Remember, we were talking about antibody structures like this. So light chains and heavy chains. This is light chain. This is heavy chain. So within the chains, if you're comparing between two individuals in IgG, you may find small difference in amino acids. So those two are called allotypy. Similar, allo means similar, typy, similar types. Small differences in amino acids. So this allotypy is what results in the polymorphism here. Very small differences in amino acid sequences could be mutations here, results in different forms of IgG in different individuals. So such an allotypy was studied by initially Grubb. So allotypy or slight variation in IgGs between individuals, slight variation in amino acids, slight variation in amino acids. In IgG among humans was described by Grubb. Okay, and further, it revealed extensive polymorphism, meaning we humans carry several different versions of IgG looks like. So there is extensive polymorphism shown with respect to IgG markers. So what happens when these different forms of IgGs are injected into us? What happens? They can show antibody response. So our immune system will recognize them as foreign particles and start making antibodies against them. That is what they have described here. They call anti-allotype response. So response to allotypes, so similar types. So our body can make antibodies against the IgGs taken from someone else. So meaning that our body is able to recognize them being different.
Sir, IgG is the antigen, right?
Now, IgG is antibody, immunoglobulin. But in this particular case, IgG can act like antigen. It's antibodies for us. But if you introduce into someone else, it may become an antigen. It becomes a foreign particle in that scenario. But otherwise, IgG is an antibody. It's an immunoglobulin, one particular type of antibody.
Sir, how will it become antigen?
How will it become? Antigen. Correct?
Yeah.
Yeah, okay. So simple. This is antibody for you. Your body is making it as an antibody, attacking an antigen. But same thing plays with in someone else's body. It is not acting like an antibody. It acts like an antigen for them. For them, it's a foreign particle. Although it is an antibody, for them, it is a foreign particle. So foreign particle is antigen. Your body makes antibodies against this antibody, which now becomes antigen here. Okay, it becomes a foreign particle. Okay. So genetics wise, let's look into the polymorphism part of it and genetic basis for this IgG. How many genes are there? How polymorphic it is? Okay, the genome allotypes, the similar types of IgG are encoded by three genes. Three genes found on chromosome 14. So GM allotypes are encoded by three genes found on the same chromosome. These three genes are called G1, G2, G3. IGH, G1, G2, G3 because they're part of the heavy chain. So Ig is immunoglobulin G, H is heavy chain, G1, G2, G3 are the forms. So you can put IgG in heavy chain, the arrow marks G1, G2, G3.
Can you repeat once?
Sure. So where do you get the IgG's variation and polymorphism? There are three genes encoding IgG. Three genes. Okay, G1, G2, G3. What part of the IgG do they encode? Heavy chain. So this is polymorphism in the heavy chain version. So heavy chain of IgG is encoded by three genes, combination of three, G1, G2, G3. Where are they present? They're present on chromosome 14. And these varieties, because you have combinations of G1, G2, G3, right? Varieties of these combinations are, how are they designated? How are they named? So they use two ways, like a numerical numbering, they can give 1, 2, 3, 4 like that, or alphanumerical. So they give alpha, beta, or letters like that. So the particular form of GM polymorphism is written with combinations of numbers and letters, alphanumerical letters. Okay. So how I'm going to show you examples here. So G1, G2, G3, gamma 1, gamma 2, gamma 3 shown here. So there are many of them, but we can only detect few of them because to detect them, we need a method, correct? We need something that can bind to it, that specifically identifies to it, something like a dye. So we have only few. So on gamma 1, we can detect 4, gamma 2, we can detect 1, gamma 3, we can detect 30. Look at them. 1A, 2X, 3F, 17Z. Like that, letters together with the numbers. So don't have to by heart them. So just have to tell that there are three forms of it, and there are enough detectable forms of IgG heavy chain polymorphism is available or is studied. So there are different forms of IgG that we can detect. How are they written? They normally go with this method of numbering them and also adding letters. So there may be different 1A, 1B, 1C like that. So there are multiple possibilities for that. So they number them like this. First, specific to specific form, G1, G2, G3, gamma 1, gamma 2, gamma 2.
How is it acting as a marker? Can you tell us?
First of all, look at this. So how is it acting like a marker? One thing that we all need to know, if there is particular sequence in a given population that varies between individuals. Okay, this is one point. So something that qualifies to be called a marker, two things that have to happen. One is variation. There must be enough variation. Second, correlation. It must correlate with something, which we'll talk about. What does it correlate with? I'm going to tell you next. But genetically, what we are talking about, it's a combination of products of genes, three genes, particularly for heavy chain, and many forms of that can be seen in different individuals. Two individuals may not have the same exact IgG sequence. Why? Because they have this different genetic regions that are being different allelic forms results in this. So how do they recognize them? They have to give some form of identification to them, then number them based on their sequence. So it is showing it is polymorphic. Now we have to ask a question, is there any correlation? in what way it can act as a genetic marker. Okay, one, population variation. So we were talking about in the previous cases, is population-wise variation is there or not? Second, is there any clinical correlation? Any disease-based correlation is there? So first we'll look into the polymorphism in various races. Okay, and then we'll talk about the connection to diseases. And then you see how it is used as a genetic marker. So different racial groups. We'll talk about in the races. Is race is a real valid concept today, but in literature it's a common thing. So still we call racial groups. So different populations, different races seems to have different versions of this IgGs. So IgG polymorphism can be specific to specific regions of the world. Like for example, Caucasians. Examples: Caucasians have GM3, 23, 5, etc. So this numbering we don't have to by heart. There's some Caucasian specific IgG version is there. So they tend to have mostly GM3 polymorphism with 23, 5, 10, 11, etc. So these are all the different number of alleles or different name of the allele. Caucasians tend to have this version, whereas Negroids tend to have GM 117, 5, 10, 11, 13, like that, with some shared elements, some different elements. So race-specific IgG can be identified. So based on the sequence of IgG, you may even identify what race they could be. Are they Caucasians or the Negroids? The technical details will be a bit more complex for this, but they have their own signature of IgGs in their body, which can be used in classifying them. In that sense, it is a genetic marker. It is acting like a reference point with respect to a particular population. Okay, but given the diversity of this, we don't have good method, good way of detecting the GM, because we need so many different reagents for it. And I don't think we have developed reagents to identify all these differences. So it is actually practically very difficult to identify the IgG markers in given population. That is a technical limitation here. Okay. So there is definitely population specific differences in IgG can be seen. Examples are given here: Caucasian specific, Negroid specific IgG can be seen. But the problem is identifying them, because we have only limited amounts of reagents to identify them. So this makes it more complicated. So we are not thoroughly using the IgG polymorphism in humans. Only to some extent we are able to use. This is indeed true for all of them. Many of them, except ABO blood groups, HLA typing, many of them are only have limited use. That is, even today they're trying to find some correlations in the recent studies. So what about the disease relevance? Is there any correlation between disease and IgG specific IgG types? So when we talked about among races, there may be a difference. Okay, so this is what these 3 points talk about, the specific variations that you find within the specific races. So differential selection, so meaning there must be some selection pressure. Why some people, some populations are actually having particular type of IgGs? Maybe there was some selection pressure acting on them. Pressure against what? Maybe some sort of specific disease or infection or any condition that they may are experiencing in the given region. Okay, so what are they? So they could be cancers. So many malignant diseases, I think some people are more and more prone to certain type of cancers because of the specific type of GM genes that they have. So there is some correlation between the IgG type and the malignant diseases, the cancers that they get. So cancers and correlation between that. This is still a very primitive, and we need further studies on this. As per some literature components, I've noticed that there is a correlation between cancers and GM genes, and plus autoimmune diseases. I was talking about this autoimmune diseases means where immune system attacks our own tissues, like Graves' disease and thyroid related problem, type 1 diabetes. So those are the examples of autoimmune diseases. So there is also some correlation between this gamma globulin genes as well as the autoimmune diseases. So they can be used as markers here. How thoroughly is a different question, but there's a correlation that can be used here employed here. And also some infectious diseases seems to have some correlation.
So we essentially have established a correlation. So whenever the feasibility is available, we can treat differentiate. That is the whole genetic marker
Exactly.
understanding.
Right. The whole goal is identify where there is enough polymorphism, and if you see it correlation between diseases, so you can use it as a genetic marker. The scientists are still in the process of establishing that connection because it is not a clear-cut connection between a disease and a polymorph. So ABO blood groups, we see some of these components, but for others, it may not be as specific as this. Correct? That is why this information is a bit confusing and bit complex. And I don't know why even UPSC have not decided to actually focus more on this, maybe lack of too much clarity on this. Okay, all right. So this is briefly about the GM markers. What are GM markers? Nature of it. Okay, polymorphism. So genetic basis of it and polymorphism and disease correlation we talked about. Okay, this is the first part of the 9.6. Now we are moving on to the second part of the 9.6 where we talk about physiological characteristics, variation in physiological characteristics, because we finished the other ones, ABO, RH blood groups, HLA, HP, transferrin, GM, blood enzymes. We finished those ones. So now we are moving on to the physiological characteristics of 9.61. So here, so physiological variation in the body, either it is gender, age variation that we have with respect to first one is hemoglobin levels. How does hemoglobin change in us? Next, body fat, pulse rate, respiratory functions, and sensory functions. Okay, so today I'm going to also cover on hemoglobin levels. Hemoglobin level. So this is something that we probably are a bit familiar about, but we are going to focus on the variations. First of all, let me explain you what is hemoglobin. Some of these you know very well. So hemoglobin is a protein, correct? Hemoglobin is a protein. And what does it do? What is the function of hemoglobin? Hemoglobin
Oxygen to
carries oxygen. It's part of RBC. It carries oxygen to various tissues of the body. Correct? This is something that you all know very well. An essential protein in our body which is responsible for carrying oxygen to various tissues. So within hemoglobin, there are two parts, heme part, globin part. So you can split hemoglobin into heme plus globin. Heme plus globin. So heme is the iron part. Heme is the iron part and globin is the protein part. If you see in the structure below here, you see the iron here? Iron is actually in the center of these polypeptides. So these, you actually see 4 units, 4 subunits together. So this is the protein part, which is globin part. Heme is the iron part. Heme is the iron part. Okay, so we don't get into the porphyrin rings, so we don't need to know so much. But basically, it is coordinated between these polypeptide chains. So an iron ring that we normally have, plus protein. And within proteins, if you see, there are 4 subunits in hemoglobin. How many subunits? 4 subunits. This is important for us to know about the polymorphism in hemoglobin. So 4 subunits. What are they? 2 alpha, 2 beta. So two chains, alpha chain, alpha chain. See the one in the bottom, pink ones, and the one on the top, brown ones. The brown ones are beta chains, pink ones are alpha chains. So alpha to beta 2, we normally write like that. Two alpha chains, two beta chains. Okay. So like that we have 4 polypeptides, 4 iron molecules, and 4 oxygens can bind to each hemoglobin. One hemoglobin can seat 4 oxygens. It's like a cab can have 4 people. A sedan, for example, or a compact car can carry 4 people. Similarly, like 4 oxygens can be seated on one hemoglobin or bound to one hemoglobin. Okay, right. So this is a brief idea about hemoglobin. Next, we'll talk about the polymorphism, genetic nature of hemoglobin. Where are the genes for it? But what did I tell you so far? Hemoglobin is an essential protein found in our body. 2 parts to it, heme part, globin parts. We can split heme plus globin. So hemoglobin, and heme is the iron, and globin is a protein part. How many different protein? How is it designed? It's a bit complex. We have 4 polypeptides, meaning we have 4 subparts or subunits that make up one hemoglobin. What are they? Alpha and beta. 2 types of them, but 4 of them, meaning 2 alphas, 2 betas. So like that, 4 together forms one hemoglobin, and iron will be the center of them in a way, and each oxygen can bind to the site where iron is present. Like that, oxygen is carried by the specially designed proteins in our body. Okay, what is the genetic basis for hemoglobin? Let's next discuss about that. So hemoglobin is definitely a complex one. As I was telling you, alphas and betas are put together. So 2 alpha, 2 beta we normally have. Okay, and there are 3 regions on our DNA which code for these parts. So 3 regions. There are multiple genes and 3 regions coding for the same protein. So chromosome 16, a region on chromosome 16, 2 different region on chromosome 11. 2 different regions on chromosome 11. Because for making all these proteins, we should have instructions being stored on the DNA, right? And the DNA should contain corresponding information for making a protein. So here on chromosome 16, and normally chromosome 16 is responsible for making alpha chain, information for alpha. Similarly, chromosome 11 has information for making beta. Like that, alpha, beta is produced by genes present on different chromosomes. They come together in the cytoplasm when proteins are made, and then they form a single hemoglobin. See how nicely it is regulated. There are different codes for it on different regions, but together the proteins come together, form one protein. 2 different polypeptides come together to form one protein. So chromosome 16 and chromosome 11 code for alpha and beta chains, and alpha beta comes together to form hemoglobin. Okay, but interestingly, you also notice gamma chains, right? So what is gamma? Sometimes it is not beta. Alpha seems to be the common form, but sometimes we have different forms of hemoglobins at various stages of our life. So we're talking about 2 things here. One, complex hemoglobin where alphas and betas come together. But interestingly, in our physiologically, in our bodies, we tend to have different forms of hemoglobins. We tend to have different forms of hemoglobins. Okay, I'll briefly explain that and I'll come back to this gamma part then. Okay. Now I'm going to talk to you about the varieties or forms of hemoglobin and how do they differ with age. So age related change here that we're talking about. And then I'll talk to you about gender related changes. So age related changes. So we have different forms of hemoglobins in our body. Okay. So There are at least 4 forms that we'll talk about here. One is 2 in adults. In adults, we have hemoglobin A, hemoglobin A2. Sometimes they are known as A1, A2. So as adults, we have A1, A2 forms. HB are hemoglobin A1, hemoglobin A2. So 2 forms. So briefly write down and I'll tell you a little bit of information on each of them. So A1, A2 in adults. So put adults, okay, to give 2 arrow marks, A1, A2 in adults. Hemoglobin is of 2 types in adults, A1 form, A2 form. The third one is in the fetus. In the fetus. In the fetal hemoglobin, it is called, it is hemoglobin F, HbF, hemoglobin F, fetal hemoglobin it is called. The fourth category called hemoglobin E or epsilon. It is not given here. Hemoglobin epsilon. E. E refers to embryo. So what did I tell you? The first 2 forms are adult forms. First 2 forms are adult forms. Second form is a fetal form. Fetus means the baby that is growing in mother's womb, embryo becoming a fetus. So fetal means before birth of the baby, it is called a fetal form. And embryo means even before fetus is formed, after zygote to embryo. When cells are dividing, we'll talk about that in unit 10. That form is called embryonic form. In embryonic form, we seems to have slightly different hemoglobin. In fetal stages, we have slightly different hemoglobin. And then as adults, we have different hemoglobins. So hemoglobin composition slightly changes with age. So 4 types, A1, A2, and F, and epsilon, or E, Greek letter E or epsilon. So we talked about this. So what is the difference? Write down the one, you write stage. So adult stage, fetal stage, embryonic stage. You write like a table. At what stage? If you already written on the left side, that's all right. Adult, fetus, embryo. Doesn't matter. Just ensure that you are writing enough information on this. Then we talk about the composition. How is it different? In all of them, alpha is common. If you see alpha 2, alpha 2, alpha 2, even in the embryo, alpha 2 is common. So alpha is same. But where does the variation happen? In the beta? Beta chain is slightly different. Because of mutations in the beta, we may carry different forms of beta chains. Okay, so what is the composition? For A1, it is alpha 2, beta 2. Write down the composition here in one column. Alpha 2, beta 2. Write down composition and alpha 2, beta. That is for A1. A2 composition is alpha 2 and delta 2. So alpha is common, right? Alpha 2 like this. Delta 2. Small delta 2. In hemoglobin F, it is alpha 2 and gamma 2. Alpha 2 and gamma 2. So 2 chains of alpha, 2 chains of gamma. What about in embryo? Can you guess letters? Epsilon. Okay, alpha 2, epsilon 2. Alpha 2, epsilon is like E, right? We write like E. E2 or epsilon 2. So, first you may ask this question. Is anything known? Why do we have different hemoglobins in embryonic stages, fetal stages, adult stages? One explanation I've seen in biology is the need for or the sensitivity. Like how sensitive? Let's say you're talking about a pregnant woman. Herself and the baby in her womb, either embryonic stage or fetal stage. Who is more sensitive? Baby or the mother? Who do you think are more sensitive? A simple question, okay, nothing complicated. Baby is more complicated. Small changes in the in the mother's body sometimes can lead to the miscarriages. Correct? So baby is sensitive. So baby should receive on priority should receive more oxygen. Otherwise, baby system cannot work. So maybe hemoglobin is designed to bind oxygen better. Let's say there is some amount of oxygen lack, not enough oxygen is present. Pregnant woman is not receiving enough oxygen, for example. A baby is designed in such a way that they can capture more oxygen. They have hemoglobin which can bind better. So in competition, if you're comparing with adult hemoglobin versus baby's hemoglobin seems to bind better to the oxygen. So maybe in nature, we tend to carry different form of hemoglobin which can bind oxygen better. So thereby protecting the baby in oxygen deficient situations. Okay, this could be one explanation here because it is very difficult to find actual answer to this in evolutionary perspectives. But what they found out here, that it could be because of the sensitivity of the baby, and baby's blood is designed in such a way that hemoglobin present in the babies can bind better to the oxygen. So more affinity or more efficiency in binding oxygen is seen. Maybe that could be the reason why when we are babies, when we are mother's womb, we have slightly different version of hemoglobin, and we are grown into adults, we have a different version of hemoglobin. And the most common in us is A1 form, which is 95%, and a small percentage of hemoglobin A2 is present in us. So in the embryonic fetal stages, most common is epsilon form, F form, but in adults, A1 is a majority form. A small percentage of A2 is there in us. Okay, so we have within us age-wise difference is there in hemoglobin. Okay, that is different from what we have talked about. So far, we talked about population-wide variation. Here we are talking about individual variation in hemoglobin. Okay, so the gene for the delta or gamma that we talked about here, hemoglobin F, this seems to be on chromosome 11. I don't know where epsilon is. It is not very well discussed in many literature sources. So here we only talked about 3 chains, alpha, beta, and gamma chains, and where the locations are. Approximately, if you know this, this is the genetic basis for creating hemoglobin. Okay. Also, there are other forms of hemoglobin. We already discussed that sickle cell anemia form, that hemoglobin S form that we know, right? So different people, based on the mutations, we have different forms of hemoglobin. Hemoglobin S form is where sickle cell trait that can be seen. We have something called hemoglobin C. So hemoglobinopathies, remember we talked about briefly? We talked about various varieties of hemoglobins. Okay, so people have different mutations. One form of the mutation that causes sickle cell anemia is called HbS, hemoglobin S. Okay, many populations, including Africans and East Indians, Middle Easterns. So I was telling you wherever malaria incidence is high, where hemoglobin S is most commonly seen. Sickle cell anemia condition this is. And hemoglobin C, there's also another type of this called hemoglobin C, which is commonly seen in West Africa. Results in mild hemolytic anemia because abnormalities in that results in destruction of RBC. So that leads to hemolytic, meaning RBCs gets destroyed. That is why anemia happens. It is not the problem of generation of RBC, but destruction of RBC is very, very high. So RBC levels goes down. It is called hemolytic anemia I was talking about. So this could happen because of a mutation leads to hemoglobin C variety. So S variety, C variety. Similarly, we also have what is called hemoglobin E variety, found in Cambodia and Southeast Asia, Malaysia, Indonesia, Vietnam. Okay, this also leads to mild hemolytic anemia and also microcytosis, or tiny, small size RBCs that these people produce. They produce small sized RBC, smaller than usual. Like that, there are different forms: S form, C form, E form. There are more than this. Okay, so we also show some variation in hemoglobin in different populations. Okay, so different mutant versions of hemoglobin is seen among human populations and within the human individuals?
Sir, has it anything to do with the ecology?
Like you mean environment, how what environment we are exposed to. So there may be a selection pressure on that. For sure, it's like this. So these people have mutations that they acquired in this particular gene, but the maintenance of this is defined by stress or maintenance by some form of selection factors. So there must be some link. Like HBS we talked about, right? Balanced polymorphism. So sickle cell trait is maintained in heterozygous condition because malarial incidence. Like that, they can have connection to malaria because these people may show protection against malaria. So that could be the selective pressure here.
I'm saying if someone is normal hemoglobin, which is common, and if he goes to some higher altitude where requirement is more, so does the hemoglobin change with that?
Yes, we're going to. I'm going to briefly talk about. Yes, for sure, ecological and ecological anthropology we are going to focus. Yes, I think you are pointing at that. So yes, we will definitely see that people who are living in high altitudes have different levels of hemoglobin. I'm going to briefly talk about that next. Okay, that is there. Ecological variation is there. There we actually manage. We'll talk about that in acclimatization and adaptation. We'll focus on that in ecological anthropology. Next unit that we are going to start mostly day after tomorrow. There I'm going to put more focus, but today I'm going to briefly mention about that. Yes, the ecological variation is also there in hemoglobin. Okay, so we show our people living in high altitude regions show increased hemoglobin levels because there are oxygen levels decrease in the high altitudes or hypoxic conditions. I'm going to tell you a case study on this in ecological anthropology. So in high altitude region, so hemoglobin levels tend to increase. This is one way of managing the hypoxia or low oxygen levels. So not only in the high altitude regions, but also in the certain lung diseases, these hemoglobin levels may increase. Certain heart problems, hemoglobin levels may increase. Even smoking may result in increased hemoglobin. Even people who are smokers, they tend to have higher hemoglobin levels because body is trying to compensate for that to pick up more oxygen, enough oxygen by having more carriers. So certain conditions, hemoglobin levels may increase. Examples, high altitude, we'll discuss more on this. Certain lung diseases and heart diseases or smoking. So you can pick up 2 or 3 examples here. Similarly, hemoglobin levels may decrease in some conditions. So any known conditions like that? So high altitude, lung disease and heart, certain lung and heart diseases or smoking can increase hemoglobin. And some other conditions like anemia, people having less RBCs, anemic conditions, they will have less hemoglobin because there is no point in pumping more hemoglobin when you are anemic. Because to match that, we normally have decreased hemoglobin can be seen, maybe defective hemoglobin, but anemic conditions, normally we notice some form of decrease in hemoglobin. Some infections also, people have decreased hemoglobin in some parasitic infections. So anemia, parasitic infections, even certain drugs reduce hemoglobin levels. Like some cancer treatments, for example, they reduce hemoglobin levels. Okay, so like that there are many. They are sufficient. Otherwise, lead poisoning, iron deficiency, copper deficiency, kidney problems. Don't need to know all of them. Few examples are enough here: anemia, parasitic infections, some drugs can decrease. So our physiological conditions, geographical locations, ecological locations where we are living, okay, physiological conditions may also define the levels of hemoglobin. So there is a variation with respect to that as well.
Sir, is it also like the cells need oxygen? So those who have less oxygen, less hemoglobin, will age slowly. than the normal people. Is there something like that?
I didn't get the question. A little bit more clear. So you said age related.
That if if someone has less hemoglobin, so
Hmm.
there would be less oxygen supply.
Hmm.
So they would age less because there is less oxygen reaching the cell.
Not necessarily. What happens to the tissues? It's like you're not supplying enough fuel to the tissues. So what happens? There is enough demand. Let's say you're walking. Unless you produce enough ATP, your cells cannot survive. What do? They have to find alternative means of energy. They start pushing your body to make more energy. Correct? So in the alternative ways, because oxygen is required for like exercise. What happens? In during exercise, you don't get enough oxygen. So what do? You use anaerobic method of ATP generation, energy generation. So thereby what happens? Your body finds alternate method. Otherwise, you cannot keep doing that job. So that is a stress for the body. So it may not work in the way that it reduces aging, but it may even adds to the aging, may add to the damage to the tissues because of the overwork. So our body has a very complex dynamics. So if you see the different examples like this, you may find it that it is actually causing more aging effect than the less aging effect of less use of it. It's like car is actually you're not fueling it, you're not using it, you just left it outside. Hasn't it aged? It has aged because of the external factors, right? It's like that. Okay. And the last one in hemoglobin I wanted to tell you is gender-wise differences, and not composition but concentration differences. Level of hemoglobin that varies with age and gender. Okay, so here our focus is on the hemoglobin part. Sometimes we also hear about hematocrit, or hematocrit is the percentage of the volume of RBC, the percentage of RBC in a given volume. So 52% of RBC in the given volume. It's like that. So this is of not relevant to us. This is basically used as a component to see is the RBC levels are less, so hemoglobin is level is less. No, it's not like that. So here we have a different hemoglobin levels that can be seen at different stages of our life and also in different genders. So at birth, we seems to have about 17 units, 17 grams per deciliter of the hemoglobin that we have. We normally use the values, right? 10, 12, 14, 17, like that. It's the same standard value. So at birth, we carry about 17, meaning we have more hemoglobin to begin with. We have higher percentage of RBC as well. More hemoglobin to begin with at birth. So not only different form of hemoglobin, but also more hemoglobin. Then at childhood, this goes down to 12. This is where people are more and more prone to anemia. Again, children in adult sense as well as in the childhood, we are more prone to anemia. If you see the objectives of the National Nutrition Mission, you will notice that children below 6 years of age, for example, they are more prone to this problem because their body is trying to or struggling to make more protein and put more muscle on it. So requires more energy. That is when you're highly active. So hemoglobin levels slightly tend to decrease here. 12 to 13 you will find here. Physiological levels. Okay. And as we grow up, in men, in adult men, we see about 16. This number may be 14 sometimes, or 16 plus or minus 2. Women tends to actually have slightly less than what hemoglobin levels found in men, especially women who are menstruating in their normal life before 45 to 55 years. 13 or 15 years to about 45 to 50 years stage where women tend to actually low or tend to be more anemic. So their hemoglobin levels are slightly less, 13 approximately you find, versus men who probably have about 16. Same women after menopause, after menstruation cycle is ended, so menopause, so they tend to have slightly more hemoglobin, about 14 plus or minus 2. But still, women tend to have less hemoglobin than men, mostly because of the menstruation process. Okay, and same with women during pregnancy also they tend to have low. So in general, women have lesser hemoglobin than men. So adult women, menstruating women, postmenopausal women, and pregnant women. So this numbers 12, 13, 14. Slightly less compared to the men. So their overall RBC level also decreases. So they're anemic. These people tend to be more anemic. So this is normal to them, but they tend to be anemic. I remember seeing some reports where they only have 5 grams of hemoglobin per deciliter, 8, 9. So which is very, very dangerous because your body's, you're starting to feel that you have shortness of breathing because your body is not able to supply oxygen. Your hemoglobin levels have fallen down. Transport is not happening. So people experience that shortness of breath. Okay, so it's very, very, they are more vulnerable in these stages of life. Childbearing ages is where they are more vulnerable, are the stages when they actually starting to menstruate. So this is a time where I think we have to focus more on taking iron rich foods. Okay, so physiologically we have differences. How do you address them? Nutritional solutions we have to find. So eating iron rich foods, that is what Indian government is promoting now. It's a very common problem. Anemia is a very common problem in India. Disease burden rate is increasing because of that. So nutritional addressing is important. So our physiological differences that we are learning can give us idea about how to tackle that problem. This is one simple scenario here. So having knowledge about this is helping us to eat particular type of food. So we can manage to get enough or pump enough hemoglobin, synthesize more hemoglobin in the body. Okay, so like that. So we talked about what is hemoglobin here, and we discussed about the hemoglobin in general, what is hemoglobin, and genetic basis of hemoglobin we talked about, and we also discussed about types of hemoglobin as we in different ages of our life, and also we talked about population differences in hemoglobin, and also we talked about the age and sex, the gender differences in hemoglobin. Okay, so there are this is a variety of information that we can have on the hemoglobin levels. Okay. Right. So next we'll move on to the body fat. I don't think we have enough time to cover that today. So tomorrow I'm going to continue with the body fat, pulse rate, respiration, and sensory systems. So there is not too much of genetics there. Too much of genetic components are not required here. The basic understanding, physiological understanding here, and any gender-based or population variation with respect to body fat levels, mainly gender variation we'll talk about, and also age variation in body fat and what parts of the body carries more fat, why. So we'll talk about the fat distribution and also both races wise or populations wise and gender wise and age wise. And then pulse rate and how does it vary? Again, age wise variation that we'll notice, and people ecological variation also we'll notice there. Respiratory function and pulse rate we're going to talk together. And finally, sensory perceptions. So how we sense things, is there a variation? Are there any genetic basis for variation? We're going to focus on that. So tomorrow we're going to wind up this 9.6. Okay. Right. Any questions? Any questions on what we discussed today? So I think I was telling you for each of these markers is slightly complex. So a brief idea, brief points on those things I was telling you. What is it? And genetic basis, polymorphism, polymorphic nature of it. Any as a genetic marker, any applications. If you focus on these 4 themes, for each of them that is sufficient. Don't stress too much. Know them, write them, just review them in case it happens to come in the exam. Otherwise, focus more on the ABO blood groups, HLA. Okay, and these ones, respiratory functions, pulse rate and respiratory functions. These are asked in the exam before. I think hemoglobin level and respiratory functions are asked in the exam. So these are the questions that came in the exam before. But otherwise, most of them are just for the sake of backup plan in case if they happen to come in the exam. Okay, because most of the questions are focused on these things. RH blood group and genetic markers in general and their usefulness they ask, and why give reasons behind blood groups being good genetic markers. So we talked about genetic basis, what is the influence of environment. I made a brief summary that I've not shared with you. For that ABO, I'm going to give a separate content and basically additional content so you can read more on it. Okay, and then discuss the role of ABO blood group system in solving paternity. So the table that I've shown you in the class, I'm going to share that with you today. So you can use that table to show how to use ABO blood groups to solve paternity issues. It's not completely solving it, but ruling out the parents who cannot be biological fathers. Okay, and write short note on respiratory function question came once, so we're going to talk about that tomorrow. And genetics of HLA and organ transplantation is already discussed. And thrifty genotype, there's one concept called thrifty genotype that when I discuss about fat metabolism, fat body fat, I'm going to briefly talk to you about thrifty genotype tomorrow. Okay, this question had come in the short note. Okay, I'll tell you very brief concept. Like any questions, otherwise, any questions on what we are discussing here? You're good? Right. So if so, we are going to have the class at 4 o'clock tomorrow. Then we are going to discuss the rest of the topics in 9.6. Okay, guys, have a good night.
Thank you, sir.
Sir, papers.
Yes, guys, I will give them. I'm almost done with it. So hopefully I can finish it tonight. So I'm aiming to discuss them in tomorrow's class. Okay, let's see after the class. Most likely I will discuss that. And before the discussion, I'll give you enough couple of hours for you to go through them. Okay, right.